The topic of this video is nucleic acid structure, and more specifically, DNA structure. So we're going to pick back up where we left off before the quiz on Friday, and we left off talking about the structure of DNA. So this first part is going to be a lot of memorization, but it's stuff that you'll really want to know, because a lot of these structural features that we're going to talk about have really big functional implications that I'm going to try to bring into this video. All right, so where did we leave off on Friday? So on Friday, we left off, we were talking about how DNA forms this really well-known double helix structure where we have two strands that wrap around each other in opposite directions. So they're anti-parallel to each other. And what that means is that if the strand on the right has its 5' end at the top of the screen and its 3' end at the bottom of the screen, then the other strand is going to be the opposite. It's going to have its 3' end at the top, 5' end at the bottom. So that's how the strands are oriented with respect to each other in the helix. We were also saying that on the inside of the helix, we have our nitrogenous bases pointing inwards, and that these stack up on top of each other like the rungs of a ladder with a distance of about 0.34 nanometers or 3.4 angstroms between them. So angstroms is just a power of 10 smaller than nanometers. And this distance ends up amounting to 10 base pairs per full 360-degree turn of the helix. So what I mean is that every time the helix goes around a 360-degree revolution, that's going to amount to 10 base pairs, okay? And within the helix, one of the things that's contributing to holding the strands together is that these bases are forming base pairing interactions with each other. So adenine base pairs with thymine, guanine base pairs with cytosine. And you're going to want to make sure that you can recognize these. So know that this top base pair is an adenine-thymine base pair, and the bottom one is a guanine-cytosine base pair by sight. And you'll also want to be able to draw these. So before we move on, let's talk a little bit about these, because I want to point something out. So notice that the two base pairs have different amounts of hydrogen bonds. Adenine-thymine has these two hydrogen bond interactions in their base pair. Base pairing is just hydrogen bonding in specific ways. And the guanine-cytosine base pair can form 3 hydrogen bonds. And so the number of hydrogen bonds that they can form is going to affect which of these base pairs is a stronger interaction, okay? But back to our double helix structure. So we were saying that our bases are on the inside of the helix, base pairing with each other, and they stack up on top of each other with a distance of 0.34 nanometers. And that ends up being 10 base pairs per turn. On the outside of the helix, we talked a bit about the inside, let's talk about the outside. We have our sugar phosphate backbone. So the sugar and the phosphate groups point out away from the center of the helix. And this should make sense from a charge repulsion point of view, because if we tried to put all those phosphate groups in the middle of the helix, we're going to have a lot of charge repulsion. So by putting them on the outside, we're really minimizing that and helping to keep the DNA structure as stable as possible. And this whole molecule has a diameter of 2 nanometers. All right, so that's kind of the basic structure of DNA. But let's look at this a little bit more closely. So let's start by, you might notice that just because of the way that the strands of the DNA wrap around each other, we have these more open regions and these more closed off regions. And this open region, there's a name for that. This is called the major groove. And you can distinguish this in a couple of ways. The first is that it's more open. The second is that what that openness actually means is that in the major groove, we can distinguish between which base pairs are interacting with each other. So can tell what the base pair is. So let's look at this diagram here. So on the left, we have adenine and thymine. On the right, we have guanine and cytosine. And so this part here, shown in the brown color, that is your major groove point of view. Like what you can see, like so you can see anything represented by this brown section from the major groove. And this yellow section, this is your minor groove. So you can see whatever's represented by this yellow line from the minor groove. So on the major groove, you can see that the identity of these base pairs becomes very clear. We have a purine with an amino group. We have a pyrimidine, the carbonyl, that is adenine and thymine. Whereas for guanine and cytosine, we have a purine with a carbonyl and a pyrimidine with an amino group. So that's one difference that we can see. We can also see that from the major groove side, the methyl on the thymine is visible, which helps distinguish the thymine from the cytosine. So in the major groove side, we can tell what that base pair is. But let's look at the minor groove side. The minor groove side, things look pretty much the same. So we have this nitrogen in the glycosidic bond. We have this nitrogen. That's the same in both. We have this carbonyl, same in both. So the only real difference that we have is in this amino group. So it becomes very hard to really distinguish with high specificity what base pair you're looking at if you're looking at it from the minor groove side. And this has big functional implications because sometimes molecules like proteins that interact with the DNA need to be sequence specific. So when you're sequence specific, you have to be able to recognize the DNA sequence in order to be able to bind correctly. So they're going to want to bind in the region of the DNA that allows them to distinguish these base pairs so that they know that they're binding to the right sequence. Whereas something more non-specific may just interact with the DNA through the phosphate groups by recognizing all those charges. So they can bind wherever they want. So this major minor groove structure is really important for how proteins bind to the DNA. All right? So that's the major and minor groove. And there's one more key feature of DNA that we need to talk about, and that is what's actually holding the double helix together. So we've talked a lot about the base pairing hydrogen bonds, and those do play a role. There's something that actually has a bigger effect. And to think about this, we have to remember that all of our bases are aromatic. So they all have these delocalized electrons that can contribute to pi stacking. And pi stacking is a weak interaction by itself. Like if we only had one of them, that would be a pretty weak interaction. But we have thousands of base pairs in our DNA molecule. So when you put all of these pi stacking interactions together, they become quite strong. So strong in high amounts, which is what we see in DNA. We have high amounts of our pi stacking interactions in DNA. So this is actually quite critical to the stability of the double helix. It's the major stabilizing force of the double helix. So pi stacking is the major force holding together the double helix. Okay? And so those are some of the key structural features of DNA. And in the next video, we'll discuss some of the key differences between DNA and RNA, now that we know kind of what the structure of DNA is. And then we'll go into a little bit about how their structures facilitate some of their functional differences.